mitosis and meiosis begins. Interphase is where it starts. In the G1 phase, the cell goes larger and larger, also organelles duplicate. Then the S phase, DNA synthesizes completely and chromosomes replicate. Lastly, G2, organelles reorganize and cells start meiosis. And now prophase 1, where chromosomes are condensed and released from the vanished nucleolus. Now they pair up gene by gene in homologous pairs forming tetrads, where DNAs cross, forming an X region called chiasmata. Microtubules binding on the kinetochores of chromosomes. Metaphase tetrads line up randomly along the metaphase plate. Each pair faces opposite poles. Now anaphase 1 begins. Homologous pairs split equally. Chromosomes move towards each pole. Everyday meiosis forms for haploid cells. Doesn't love and meiosis, meiosis, osis, osis. Doesn't love and meiosis, meiosis, osis, osis. Doesn't love and meiosis, meiosis, osis, osis. Doesn't love and meiosis. Everyday meiosis goes on and on, along with checkpoints. Now telophase to nuclei forms enclosing chromosomes. Then the fibers break up, cytoplasm next in cytokinesis. Animals form cleavage furrows and plant cell plates, then cells divide into two. If non-disjunction occurred and cells divided, trisomy 13 results once fertilized. Now we go to meiosis 2. Unlike meiosis 1, there's no interphase, similar to mitosis, but diploid cells don't exist. Instead, haploid cells exist, that are different from the parent cell, formed in the end. Now we go to prophase 2.
A mitotic division of each haploid cell form in meiosis 1, similar to prophase 1, but chromosomes don't pair up, also genes don't cross over, only half of the chromatids are present from prophase 1 in prophase 2. Chromosomes line up separately along the metaphase plane. This is metaphase number 2, and a phase 2 begins with sister chromatids are separating and pulled to the opposite poles of the cell. Motor proteins drive the process. Session lasts with meiosis 2, meiosis, osis, osis. Session lasts with mitosis, mitosis, tosis, tosis. Session lasts with meiosis, meiosis, osis, osis. Session lasts with meiosis. Everyday meiosis goes on and on, along with the checkpoints too. Sister chromatids are formed and moved away oppositely, become sister chromosomes. To daughter self-give rise, produce for genetically different haploid cell. Spindle disenvelope, nuclear envelope reform. Regulators and checkpoints ensure nothing can go wrong. G1 check growth factor size nutrient to decide if cells should divide. G2 checks DNA damage and cohesion of the process. Spindle checks if chromosomes attach to the spindle at a metaphase plate. Cyclins drive the events of the cell cycle, binds with CDK. MPF is an example of cyclins and CDKs works together. Checkpoints prevent mutations. Meiosis is what cells go through. It gives us variations. Mitosis is what cells do. They make us grow and develop. Everyday cells divide, make me and you. This is cell division.
Do it again. No, I'm trying to zoom. Shaking? I'm trying to zoom. Your zoom stops at 1.5. What? Right now? Oh.
Yes. Go ahead.
You should be. Wait.
Go ahead. down.
Who are you recording it for?
Myself. We need another card for
Questionable.
this. Every day meiosis go on.
Can we duplicate?
Oh, like
The social history of others.
There's just so much going on.
Profanities, foul language, ethnic slurs, and English-speaking world. Nice.
Guys, Robin's starting his photography career. Please. They're doing the JYP. No! They're doing the JYP song. Later.
3, 2, 1, go. Writing. Scream!
Oh, are you okay? Oh, no. Don't worry. I got you.